Hello friends, welcome to Z Tutors. Today we will be discussing about the testis. In the previous video, we had studied a few topics like the male accessory glands, the male accessory ducts, the testis. We just had an overview, but in this topic, we will be studying about the internal structure of the testis, the composition of the testis, and the function of the testis, and so on. Okay, so let's begin. So as you can see, the testis is oval in shape and the length is about 4 to 5 centimeter and the width is about 2 to 3 centimeter width-wise. Okay, and the testis is covered by an external dense covering. So what is this covering called? This is nothing but the scrotum. What does it do? The scrotum basically is for protection. Apart from protection, the scrotum helps in maintaining a certain temperature, a preferable temperature that is necessary for the process of spermatogenesis. What is spermatogenesis? It is the process of sperm formation. Okay, so what does the scrotum do? It maintains a temperature that is about 2 to 3 degrees Celsius lower than the internal body temperature. And why does it do so? Because a preferable temperature is needed for the process of sperm formation. I hope this is clear. Okay, so this point is clear. Next, each testis has about 250 compartments. So these black lobules, lobe-like structures that you see, these are called the testicular lobules. Okay, and each testis has about 250 compartments, each testis. Okay, and inside these compartments are present thin, highly coiled structures. Okay, these highly coiled structures are known as seminiferous tubules. So basically these seminiferous tubules are the sites for sperm formation. Okay, this is where the sperms are produced. So each lobule, each black lobule consists about 1 to 3 highly coiled structures. Okay, so we will be studying about the sperm formation in detail in a topic called spermatogenesis in the later slide. Okay, now this seminiferous tubules, if you take a section of a seminiferous tubule, these are tubules, right? So we take a section. So these are lined by two types of cells. Okay, one is Sertoli cells, and second one is germ cells, male germ cells. These are germ cells are also called as spermatogonia. Okay, so the seminiferous tubule is lined by two types of cells. So this cuboidal cells that you see, these are the, I'm just giving you a rough idea. Okay, these are the germ cells, and the Sertoli cells are present in between the germ cells like these. Okay, so the job of the Sertoli cells is to provide nourishment, and the germ cells form the sperm through various intermediate processes. Okay, so now we will be seeing what are male sex accessory ducts and male accessory glands. The male accessory ducts include retitestis, vasa efferentia, epididymis, and vas deferens. Okay, so let's not get confused about all these ducts. Okay, we will be going slowly to understand better. Okay, so the seminiferous tubules of the testis open into the vasa efferentia through retitestis. Let's not get confused. Let's not get confused. It's extremely easy. You just have to understand. Okay, I have just included a picture here which is not really clear, but for easy understanding. So now we have studied about the seminiferous tubules. These are the seminiferous tubules. Now what are they saying? That the seminiferous tubules open into the vasa efferentia. Okay, so now these channels that are emerging like these, so okay, that are emerging like these are the vasa efferentia. Okay, now the seminiferous tubules open into the vasa efferentia through retitestis. Now this net-like structure that you're seeing here, these net-like structures, these are the retitestis. Okay, and these channels that emerge from the retitestis are the vasa efferentia. Now this point is clear. Now the vasa efferentia is leaving the testis. This is the testis. The vasa efferentia leaves the testis and joins the epididymis. So this entire structure that you're seeing here, this entire structure is the epididymis. I'll be showing you a video where it will be more clear for you to understand. Okay, the vasa efferentia leaves the testis and joins the epididymis. Okay, So where is this epididymis located? This is present along the posterior surface of the testis, of each testis. Okay, so another point is clear. Now where does this epididymis lead to? The epididymis then leads to the vas deferens. This thin ducts that you see, tubes that you see, is the vas deferens. So seminiferous tubules to the vasa efferentia through the retitestis. Okay, seminiferous tubules. Second one is retitestis. Okay, then vasa efferentia. Then we have epididymis. Now we have vas deferens. Okay. Now the vas deferens. Okay. Let's refer to this picture. The vas deferens loops over the urinary bladder. So this structure that you're seeing is the urinary bladder. Okay. And these tubes are the ureter. Okay, don't get confused between the ureter and the urethra. Okay. Ureter collects urine from the kidney. Okay. Urethra is present in the penis. It's a common passage for the semen and urine. Okay. So the vas deferens loops over the urinary bladder, and these two wing-like structures that you're seeing here is the seminal vesicle. The vas deferens connects with a duct from the seminal vesicle and forms a common passage, that is the ejaculatory duct. So now if you refer at this picture, you will see that this is a duct from the seminal vesicle and this is the vas deferens. Okay, these two, the duct from the seminal vesicle and the vas deferens actually merge to form the ejaculatory duct. Okay, it's clearer in this picture. Now the ejaculatory ducts, one on each sides actually, they merge to form the urethra, a common passage. Okay, I will be showing you a video later which will make it more clearer. Now what do these ducts do? These ducts, they store and transport the sperms from the testis to the outside through this common passage called urethra. Okay, so this entire structure that you're seeing here is the urethra, and this opening is called the urethral meatus. What is it called? Urethral meatus. Now, this elongated structure that you're seeing is the penis. Okay, and this enlarged portion towards the end of the penis, this bulbous structure that you're seeing, is the glans penis. Okay, now this glans penis has a loose fold of skin over it. It has a loose fold of skin over it. This is called as the foreskin. I hope it's clear. So all these points are clear. This one is clear. Okay, so up to here we are clear with the male accessory ducts. Next we will be speaking about male accessory glands, that is the seminal vesicle, the prostate glands and the bulbourethral glands. We will be speaking about it in a separate video. Okay. So another important thing here is about the seminal plasma. What is the seminal plasma? Seminal plasma is nothing but the fluids from the epididymis, the vas deferens, the seminal vesicle, the prostate. These together, the secretions of these basically constitute the seminal plasma, and the seminal plasma mixes with the sperm to form semen. I hope it's clear. Okay. So in the next video, okay, before that, let me show you a video for clear understanding of the male reproductive structure. You can have a look. Yeah. Okay, so yeah, the portion has gone below the slide. Anyway, this is the testis, and these structures are the epididymis, this is the vas deferens, and it loops over the urinary bladder. So this one is the urinary bladder, and these wing-like structures that you're seeing is the seminal vesicle. Okay, now as you can see here clearly, the duct from the seminal vesicle and the vas deferens, they actually converge to form a common passage, that is the ejaculatory duct. Okay, now these ejaculatory ducts join the urethra, and this one that you're seeing is the prostate gland. Okay, so actually below these prostate glands, bulbous structures are present, that is the bulbourethral gland. So from this video, it's clear where the organs are actually present. Okay, so that's it for today. If you like the video, don't forget to like, share, and follow. Thank you for watching Z Tutors. See you next time.